Hey everybody, welcome to another lecture video, this time on lipids. So we're gonna look at some different classifications of lipids. We're not going to cover all of them. In fact, we're not going to talk about waxes, for example, at all. Waxes are lipids. But we are going to talk about some biologically relevant ones, and then we will transition into a more of a discussion on biological membranes. And so, before we can talk about membranes, we have to talk about the components of them, and those, of course, are um, the lipids. And so, that's where we'll start. All right, so lipids are a diverse class of naturally occurring organic compounds. They all share the same characteristic, at least in that a portion of them is hydrophobic, so water hating. They play many roles in cells. Energy storage, such as fats and oils. They are constituents of membranes, such as glycerophospholipids, sphingolipids, cholesterol. They are hormones, steroid hormones. We call those glucohormones. We'll get the name on there again later when we get to that. And there are mineral corticoids, or I always forget the name of that one too. We'll talk about those as well. Two different kind of hormones. Hormones that affect water flow are the mineral ones. Again, steroids are the commonality. Vitamins, fat-soluble vitamins tend to be very lipid-like. And we'll talk about a couple here. Maybe vitamin A, maybe a little bit of vitamin D, or maybe I'll just have you guys read about them in the book, depending on how much time we have. And they can be oxygen or electron-carrying molecules like heme. These are also lipids. So we'll talk about a few of these different classes of lipids. Now, for lipids that are very hydrophobic, such as fats and oils, movement and storage of these lipids in the aqueous environment of the body requires special structures. We'll talk a little bit about those. Other amphipathic lipids, such as glycerophospholipids, that have more of a polar region and a non-polar region, excuse me, can actually spontaneously organize themselves into lipid bilayers. And so we'll look at those and how that happens in water. And another sort of interesting thing about many of the lipids in our bodies is that they can all be derived from acetyl-CoA. Acetyl-CoA is it's coenzyme A is a coenzyme, and acetyl is a carbon or a two-carbon group like this that can be attached to coenzyme A. And we call this acetyl-CoA. Acetyl-CoA is a metabolic intermediate. We can actually make fats and break down fats from this molecule. So it's just a neat that many of the fats that we can make all kind of come from this thing, from this molecule. Okay, so they are insoluble in water. They're amphipathic in nature. Let me go ahead and clear this. And lots of different diverse roles. Now, there are open chain lipids and there are closed chain lipids, and so that's kind of why I'm showing you these pictures here again. So open chain lipids, such as this phospholipid, we know it's a phospholipid because it has a phosphoric acid group embedded in it. And these include fatty acids, triacylglycerides, or triacylglycerols, or triglycerides. They've kind of gone through an evolution in terms of their names. Sphingolipids, phospholipids, glycolipids. We'll talk about all of these kind of individually. I mentioned some of your lipid-soluble vitamins, vitamin A, D, E, K, and then some other molecules called prostaglandins, leukotrienes, thromboxans. We'll look at some of those as well. These are all open chain or linear lipids. Then we have cyclic lipids, and these include cholesterol, which is this guy. We'll talk a little bit more about cholesterol and its transport and storage, and then some of the negative of cholesterol in a little bit. And then steroid hormones and bile acids are also in this category. Okay, so we have quite a few types of lipids. Now, of the open chain lipids, we're going to look at some fatty acids, and you can see fatty acids are actually components in several of these different types of lipids. In fact, it looks like they're in all of them. So our simple lipids are going to be a glycerol molecule and some fatty acids. We're going to spend a little bit of time talking about different fatty acids first, and then we'll kind of come back and tie this all together. As they get more complex, we'll see that in this glycerol molecule here that has some positions that some fatty acids can add on to, if we start to substitute one of those fatty acids for other things, such as a phosphate, some amino alcohols, or different backbone molecules, so in this case, sphingosine, as opposed to glycerol, these subtle differences and variations are going to give rise to all the different kinds of lipids that we are going to talk about, and we're not going to go in depth into all of the different functions of these lipids, but we'll see that there is a purpose to some of these differences. Okay, so let's start with fatty acids. So coming back to this image here. Actually, I didn't show you this one before, so this is new. You guys haven't seen this before. The most ubiquitous lipid, the most commonly found lipids, not just lipids in the cells, are the fatty acids. Now, they're found in the fats and the phospholipids and the sphingolipids. They often serve as membrane anchors for proteins and other biomolecules. So they're held on to the membrane through these fatty acids. They're important for energy storage. We'll see that in a little bit too. Actually, I don't have any images of fat cells, but we'll talk about that very briefly. We store energy as fat and as these fatty acids. They're important in membrane structures, precursors of all the lipids, or most classes of lipids. Okay, you can see from these two images here of lipids that they're characterized by basically a polar head group, and this is the polar head group that we're talking about. It's polar because if you remember your organic chemistry here, or even your Gen Chem here, there's a very electronegative atom, oxygen, attached to this carbon, creating some little partial positives and partial negatives. So this carbonyl on its own is already charged, and then we've got this little bit right here too. This is an acidic hydrogen. This is going to come off and leave some negative charges. At most physiological pHs, this will not be a carboxylic acid, but will be a carboxylate ion. We'll talk a little bit about that too. And so there's lots of charges on this part of the group. Long hydrocarbon tail as well. Now, fatty acids with hydrocarbon tails that lack any double bonds are classified as saturated. You might remember that. And that's because every carbon has the maximum number of hydrogens, and those with one or more double bonds in their tails are known as unsaturated. And the effect of the double bond on a fatty acid is actually to introduce a kink or a bend. And you can see that here in oleic acid. Now, multiple bends and kinks give rise to lots of crazy little circular structures. Arachidonic acid, for example, a 20-carbon acid, has like 4 different double bonds and then polyunsaturated, as opposed to just what we would call oleic acid, a monounsaturated. Anyways, so that's a little bit of a difference there. Now, stearic acid is a saturated fatty acid. It has a straight hydrocarbon tail, and you can see in this little table off to the side some of the most common saturated, and then over here unsaturated fatty acids. We'll see and actually right here we can see that fatty acids with unsaturated tails, and so what I'm showing you here is unsaturated versus saturated. Unsaturated tails have much lower melting temperatures than those with saturated tails. So the shorter tails, size also matters too. So if we go, just by looking at the different lengths of these carbons. The 12 carbon fatty acid melts at 44 degrees, whereas the 20 at 77. So having less carbons means a lower melting point, but also introducing a double bond. So let's take a look at palmitic acid, 16 carbons. We go here to palmitoleic acid, 16 carbons. The notation for double bonds is to show how many carbons you have and then how many double bonds you have. So palmitoleic acid only has one double bond, so it's a 16-1. It's a delta 9, so that means, oh, I'll show you what that means in a second. But that's the position of the double bond. Look at the melting point. Negative 0.5 Celsius compared to 63. That one double bond changes the melting point dramatically. Now these are the differences essentially between fats and oils. Fats are going to have a higher content of saturated fats. Oils are going to have a higher content of unsaturated fats. Fats with more than one double bond are called polyunsaturated. Now plants are actually an excellent source of unsaturated and polyunsaturated fats. Let's see. Double bonds that are found in the fatty acids are predominantly in the cis configuration. This means, well, when we have our double bonds, they can either be like that, or they can be like that. Cis means that the two groups are on the same side, and trans means that the two groups are on opposite sides. If you were to split the double bond in half and see where everything is laying, if your branches are on opposite sides of that plane, we call that trans. And so most double bonds are cis. Now, there is some trans fats. Let's talk about how those form. So there's a process called hydrogenation, where we reduce the double bonds and make saturated hydrocarbons. They do this. They do partial hydrogenation. I should actually put partial here, because this is when it happens. When we partially hydrogenate something, think about peanut oil being partially hydrogenated so that it's more solid, to get peanut butter, or butter, canola oil partially hydrogenated to make margarine spread or some kind of spread. The process actually makes trans fats. Trans fats still pack together, just like the saturated fats do. They still have the same zigzaggy shape, but then they do still have that double bond. And then later in our bodies, we're going to see that having the trans fats actually leads to a raised level of our LDL, and actually lowers the levels of our HDL also. We'll talk about what that means in a little bit. Let's see. So I told you guys about saturated versus unsaturated, about the double bonds basically. Now the reason, right, that the saturated ones have higher melting points is because when their hydrocarbon chains, right, packed together, lots of these guys are near each other. There's interactions. We call them van der Waals forces, hydrophobic forces. They hold these guys together. If this had a kink in it, if we had two, let's say, fatty acids that had kinks in them, something like, that's an over dramatic one, but some double bonds like that, you can imagine two fatty acids are going to have a hard time lining up with each other in order to have enough, interactions with each other that they're going to hold on to each other with any kind of energy. And so think about Pringles, right? If Pringles in a can, they're all the same shape, so they all pack together really nicely. You can get a lot of them in there. That's what saturated fats do. And then think about like some kind of other, I don't know, chip that doesn't do that. Popcorn, for example. You can't, popcorn's going to take up a lot of space. They're bulky. You can't fit them in. You don't get as many inside of a package. So that's kind of what's happening here. Okay, and so then showed you guys the difference between the effects of what happens when we do hydrogenation. Okay, let's talk about numbering. So whenever we want to describe an unsaturated bond, we have two options. We can use the delta system, or we can use the omega system. Now, you're more familiar with the omega system because you've heard of omega-3 fatty acids and omega-6 fatty acids and maybe omega-9 fatty acids. So omega-3, omega-6 means that the fatty acid, the unsaturated bond, the double bond, starts at from the omega side, meaning the tail end, the number 1, the number 2, the number 3 carbon, 4, 5, the number 6 carbon, 7, 8, the number 9 carbon, and so on. So those are the omegas. When you're talking about the delta fatty acids, then those are from the carboxyl side, the C double bond O side. Now these guys are a little less common. We don't hear this as often, but they would be described as, like, let's say delta 9. Oh, and actually we were looking at one of those earlier. Let me jump back. Where was our delta 9? Right here. So palmitoleic acid is a delta 9. So that tells us where the position of the double bond is. On the 9th carbon going this way on that 16 carbon compound. So fatty acids are described as being essential fatty acids if they must be in the diet. So we can't make them ourselves, similar to the amino acid definition. And the non-essential fatty acids, if we can synthesize them. Now humans and other animals lack the certain enzymes to make the double bonds at positions greater than delta 9, so to make these guys. So fatty acids with double bond beyond this position must be obtained in our diet. So linoleic acid and linolenic acid are both in this category. Let's see if we have an actual image of linoleic and linolenic. So both of these are ones that are essential that we would want to get in our diets. Now, if we could get these in our diets, we can make essentially all the other ones from that. So all the related unsaturated fatty acids we can make once we get those into our diet. We don't need to also consume others. Let's see. So the omega-6 and the omega-5, omega-6 and omega-3 are linoleic and linolenic. So if you've ever heard that you need to, consume more omega-6 fatty acids or omega-3 fatty acids, this is why they're good for us. Now, omega-6s also have some kind of pro-inflammatory effects as well, so there's an added bonus to consuming those. All right. So this is a fat. This is an oil. We call these triglycerides. Triglycerides or triacyl glycerols or triacylglycerides. They come from the fact that there's a glycerol, so it is CH2, CH2. Okay, and a fatty acid, and the fatty acid is these guys. There's 3 of them, so the tri comes from that. They can be different fatty acids. They don't have to be the same. They're often not the same. In different plants, the differences in their oils is the fact that they have differences in which fatty acids are attached. Okay, so a little bit about the reaction that happens here. This is an ester reaction, so an esterification that happens between an alcohol and a carboxylic acid. And this being the rest of the fatty acid. When these guys react together, we get a nucleophilic attack on the carbonyl carbon, similar to what we saw in the method of action, right, with the serine proteases in our enzyme chapter. And then we end up with an ester bond and a loss of a water molecule. So these can be hydrolyzed, meaning cut with water, back into these constituent kind of pieces. Okay, so fats and oils are the primary energy storage for animals and for us included. They're a bunch of names, we talked about that. So They have the same basic structure, regardless of if they come from plants or from animals. It's just the fats that are attached to them that are different. Biological oils are not the same as petroleum-based oils or like oil from the ground. Okay, that's not the same. So, increasing the number of unsaturated fatty acids actually decreases the melting temperature. So, like organisms like fish that live in cool environments actually have fats with more unsaturation um, to better cope with their environments. This is why fish oil tends to contain a lot of those polyunsaturated fats that we want. Now, fats are stored in specialized cells called adipocytes. Adipocytes. And we have enzymes called lipases that actually go and release fatty acids from these fat cells in those hydrolysis type reactions. I think triacylglycerol lipase might be one of those. And this enzyme goes and cleaves the first two fatty acids from the fat, and then a second enzyme, monoacylglycerol lipase, comes in and cleaves the last one. And then, excuse me, and then they can go and be available for energy production, or they can go and be converted into, let's say, other forms of these acids, or other forms of these lipids. All right, let's see, what do I want to talk about next? Let's talk about how we make soap. Soap's interesting. So let me get rid of. So saponification is the technical term for the base hydrolysis of a fatty acid, or of a triglyceride. There's two ways to undo this ester bond, and one is an acid catalyzed reaction, the other is a base catalyzed reaction. The acid catalyzed reaction gives us back our fatty acids, right? Because hydrogen ions go back and get added onto the carboxylate ion right here. And so then we get our fatty acids back. But if we do this in base, then all of these don't get added back on because there are none. They all got neutralized by the sodium hydroxide. And so instead we end up with the salt of these fatty acids. We get back our glycerol, but we get back what we call the salts. And so sodium salts, potassium salts, you can use different ions for this, and they'll give you soft soaps and hard soaps and whatnot. But these soaps look like this. And so the reason that we use molecules like this for cleaning is because we often are using water to rinse off stuff that doesn't like water, and they don't mix, and so they don't wash. But if we include a molecule that helps to bridge the differences in their polarities, water can very easily associate with part of the soap molecule, and the dirt or the grease or whatever it is associates with the other part of the soap molecule, and then together they're washed away. And so that's what soap does. Now, if we put a bunch of soap molecules into a solution, they actually will spontaneously form into these neat little structures called micelles. And a micelle is thought of as maybe a precursor to what cells look like now, right? Lipid bilayers, well, we'll see what they look like now in a little bit. But more complicated than this, but you can see they're little compartments. All of the nonpolar tails tend to organize on the inside, similar to how the nonpolar R groups on the amino acids tend to clump on the inside. And so we get these little enclosed compartments. Of course, nonpolar in, polar out. We want cells that are going to be able to have, polar inside, polar outside, and then, nonpolar in the middle. That way we can move stuff in and out. Things can happen inside, things can happen outside. So we need a more sophisticated system or picture. All right, so that's soap. That's how soap works. Let's switch gears now and talk about glycerophospholipid. So a glycerophospholipid is an important component of the lipid bilayer. Now, phosphoglycerides, as they're also known, are structurally related to fats. So let me write that here. Glycerophospholipid. lipids or phosphoglycerides. Okay. You can see that they have two fats in the normal positions, but instead of having one of those fats, we have a phosphatidic acid that has formed a phosphoester bond with that glycerol. And so this turns it into a phospholipid. Now, there's another component. We're going to talk about that right now. So this phosphoric acid is a simple glycerophospholipid that usually gets converted into phosphatidyl compounds. And these are made by esterifying basically various groups such as ethanolamine, serine, choline, and inositol, to name a few, to the phosphate of the phosphatidic acid. So phosphatidic acid, phospholipid, has everything on it. So all of these compounds are found in the lipid bilayer in aqueous solution due to the fact that they have kind of a, you can see another structure here showing the same thing. This is glycerol. So we've got the glycerol backbone right here. We've got the 3 ester bonds and the phosphoester bond. So this is phosphatidic acid. And then when it gets added to one of these guys, we get the alcohol, these different alcohols. We get the various different phospholipids. So also mentioned here, the fatty acids don't have to be the same as each other. Okay, let's talk a little bit about the different phospholipids. We won't name all of them. Just go through a couple. So there's the phosphatidyl ethanolamines. So when this guy gets added on, so phosphatidyl ethanolamines are found in all living cells. They're one of the most common phosphatides, making up like 25%. They're common constituents of brain tissue and spinal cord, making up as much as 45% of all the phospholipids found in those membranes. These phosphatidyl ethanolamines are asymmetrically distributed across the membrane, meaning that we find them more on the inside of the bilayer, facing the inside of the cell. We call it the inner leaflet. Then on the outside, we see more of them on the inside. Now, metabolically, these phosphatidyl ethanolamines are precursors of phosphatidylcholines. So you got to make this guy before you make this guy. So phosphatidylserines, these guys down here, are another group of phosphatidyl compounds that are also preferentially distributed across the bilayer. Like the phosphatidyl ethanolamines, the phosphatidylserines are found on the inside, and when apoptosis, which is like a cell suicide, happens, the preferential distribution is lost, and the phosphatidylserines actually start to show up on the outside of the cell, and those are markers for macrophages to actually bind to them and destroy the cells. So they serve signaling functions as well as being part of the backbone, part of the membrane bilayer for their charges. We'll talk about how their charges matter too. Phosphatidylcholines, they're more commonly found on the outer leaflet, so kind of facing like the blood and the lymph fluids, other tissues. These compounds, you can get them from eggs and soybeans. They get, let's see. We don't need to. I don't even have a picture to show you about how they're kind of shown or distributed, at least not at this point. I've got this image. So here, this one's actually a little bit better. This one shows how we can kind of picture the hydrophilic part and the hydrophobic parts together. So in the bilayer, the hydrophobic part, so facing either the inside of the cell or the outside of the cell, will be these guys. And then all of our fatty acids tend to clump together over here. Now, the ethanolamine or the choline or the serine or the inositol get added on to the phosphate, So they are these R groups shown here up at the top, and you can see what those structures look like if this P right here was attached to this O or this O or this O. What does this PI look like? This inositol, phosphoinositol. Kind of looks like a sugar, doesn't it? We can talk about inositol actually very quickly because inositol itself is not a lipid, but it is found in many lipids. And it has a taste that's like half as sweet as sucrose, just to kind of point that out. Okay. I know that we're going to talk about sphingolipids here in a second. Yeah, I guess sooner than later. Okay. So those are our phospholipids, or our glycerophospholipids, or what was the other word? Phosphoglycerides. Okay, again, glycerides or glycero because of the glycerol backbone. Now we're going to switch gears and look at the sphingosine backbone. There's another one. So here's how that differentiates or looks different. So we've got sphingosine. Now let's take a look at sphingosine. So sphingosine's right here. Sphingosine is much longer, right, than glycerol. Glycerol had 3 OHs on it. This one only has 2, but it also has like a built-in fatty acid already. So it's often shown like this, with a big old long fatty acid part on the sphingosine. Now, of course, there is room to add another fatty acid, so what I just drew over here actually doesn't make any sense. Let me get rid of that. Sorry. Sometimes what I was trying to get at is they show sphingosine like this. And they say, this is sphingosine, and then here's a fatty acid, and then here's this stuff, you know. So to kind of show it in the same, so what you end up looking at is you're like, oh, the fact that it's got these 2 long parts is almost just like what glycerol has, and it is just like that. So so here's sphingosine. So sphingosine is structurally different than the glycerol. It's similar to the phospholipid in the fact that it has like 2 acyl chains, 2 big long hydrocarbon chains, and it's got a polar head group. Okay, sphingosine is the backbone though; it's not glycerol. So let's talk a little bit about some other terms. So before we get into this one real bad, let's talk about lecithins. Lecithin. A lecithin is a generic term for a combination of lipids altogether. They include having phosphoric acids, glycerols, glycolipids, triglycerides, phospholipids, all of these mixed together, and they're actually what we call wetting agents. They help with emulsifying and encapsulation. They're used as anti-sludge additives in motor lubricants. It's used in candy bars to keep cocoa and cocoa butter from separating. Kind of like soap. It's like an edible soap. So we like lecithins for that. Now, in the body, they, I don't know if they have a function in the body other than we call lecithins combinations of these other molecules that actually do have roles in the body, right? Triglycerides, phospholipids. So safe as a food ingredient. Let's see. They actually do have some kind of health concern, though. I think some of the gut bacteria can actually metabolize them into something else that might contribute to cholesterol deposition and stuff, or atherosclerosis, is it? Okay, sphingolipids have sphingosine. Now, fatty acids are also components of this branch of molecules called sphingolipids. So that's why they are all grouped together. These are the complex lipids as opposed to those simple ones that just had fatty acids. These these sphingolipids, though similar in structure to the glycerophospholipids, are synthesized completely independent of them, starting with palmitic acid and amino acid serine. Now, sphingolipids are named for the amino alcohol sphingosine, though they're not synthesized from it. It is part of the molecule. Now, if the R group, and so the R group here that we're going to talk about, let's see, do I have a picture of that? Okay, so let me show you what R group we're talking about. So in sphingosine, sphingosine backbone is going to have a fatty acid attached through an amide bond. Actually, that's not an amide bond, that's an amine bond. And then we're going to have these 2 hydroxyl groups. one of these hydroxyl groups actually can form an ester bond. And so if this is a hydrogen, which it is right here, it's a hydrogen. I erased the hydrogen by covering it with my red, but you, you should still be able to remember that that is a hydrogen. Okay, there's a hydrogen there on that O. When the R group is a hydrogen, the molecule is called a ceramide. Okay, there it is. When the R group is phosphoethanolamine, so, this guy, the phosphate and the amino alcohol ethanolamine. So when that is all attached, so this is a picture of that, by the way. When that is on there instead, the molecule is called sphingosine. I'm sorry, sphingomyelin. Sphingomyelin. This is an important component of the myelin sheath and in lipid membranes. This is also the molecule that gets degraded if you have MS, multiple sclerosis, autoimmune disease, right, that attacks your myelin sheath, degrades it. That makes your nerve transmissions, your nerve cells, your action potentials not be as long, and your signals don't go as far, and you get all kinds of issues from that. Okay. If a single simple sugar is added, single simple sugar, instead of phosphoethanolamine, or instead of being a hydrogen, then we actually call that a cerebroside. So this picture is showing you sphingomyelin, where, so I picked the wrong OH. It doesn't really matter. Here's the other OH. That's the right OH. What I mean is, when I was showing you this OH, should have been doing it on that oxygen. So sphingomyelin, it's called a cerebroside if it's got a sugar, simple sugar on it, and it's called a ganglioside if it's got an oligosaccharide on it. Remember from the last lecture, oligosaccharides are just, many sugar molecule monomers linked together. Okay, so these complex sphingolipids play roles in cellular recognition and signaling. Sphingolipids are found most abundantly in the plasma membrane, and are almost completely absent from mitochondrial and endoplasmic reticulum membranes. So not anywhere on the insides of cell membranes, just on the outsides. I mean, on the the border, the outside of the cell. I don't mean on the outside membrane of the cell, the outside leaf. I just mean found in outside membranes, because what maybe you don't remember from chapter 1 if you didn't read it, but if you did read it, you remember that the cell also contains things called organelles, right? Mitochondria or organelles. They have membranes, and organelles have membranes, and our whole cell has membranes. Well, this is where we find these guys, in our cell's outer membranes, but not within the membranes of the internal organelles. That's what I'm saying. All right, in animals, dietary sphingolipids have actually been linked to reduced colon cancer, reductions in LDL, increases in HDL. These are transporters of different lipids in our bodies. All right, let's see. I've got some variations of some fatty acids here I want to talk about as well. So first are the eicosanoids. I never know if I say that right, so I'm going to write it out for you. Eicosanoids. Now, fatty acids that are made from omega-3 and omega-6 fatty acids have these 20 carbons, and they include one called arachidonic acid. And this is arachidonic acid that I'm actually showing right here. Now, arachidonic acid has a couple of double bonds in it. Now, it can form a class of compounds called eicosanoids by basically being oxidized. So oxidation of that acid leads to eicosanoids. These are both arachidonic acid, by the way. One straight chain and one kind of showed bent. Now, eicosanoids also have subclasses, including prostaglandins, prostacyclins, thromboxans, lipoxins, leukotrienes, and endocannabinoids. We're going to talk about some of those. Now, these eicosanoids play important roles in inflammation, immunity, mood, behavior. Okay, so most of these that are down here are synthesized from arachidonic acid, okay? And they are generally referred to as eicosanoids. Let's talk about some of the classifications. So we've got prostaglandins. Now, so prostaglandins are a collection of molecules that kind of act like hormones. They're derived from the arachidonic acid, many differing, even conflicting physiological effects. Constriction or dilation of vascular smooth muscle cells, induction of labor, regulation of inflammation, thermo, they thermoregulate the hypothalamus to induce fever. They are so. These are two prostaglandins. This is prostaglandin PGH2. Not that it matters. PGH2. This is prostaglandin E. I'm just going to say prostaglandin E. I was going to say something and I totally lost what I was going to say. Was it about thromboxins? Anyways, okay. So prostaglandin, what is PGH? Let's talk about some of the interesting ones. Not necessarily these two that I have pictures of, but like PGD2 inhibits hair follicle growth. It's a vasodilator, causes bronchial constriction, which is actually more affected by or in asthmatics than non-asthmatics. PGE2, which is the one I've actually got shown here, exerts effects in labor, stimulates bone reabsorption by osteoclasts, induces fever. It's a vasodilator. Anyways, does lots of things. Some of them actually have opposite effects, which is funny. There's I don't remember which prostaglandin it is. It might be 2 alpha. Either way, when it binds to a certain receptor, let's call it the EP1 receptor, it causes bronchoconstriction and smooth muscle contraction. But if the same molecule binds to the EP2 receptor, it causes bronchial dilation, which is the opposite of bronchoconstriction, and it causes smooth muscle relaxation, as opposed to contraction. So competing opposite effects. Again, sometimes also prostaglandins are formed as like trauma or as an inflammation. So when you get injured, these prostaglandins are produced, and those are the signal for pain. When you take things like non-steroidal anti-inflammatory medications, these are often blocking the conversion of the arachidonic acid into these prostaglandins. So that's how those medicine like Advil or ibuprofen work, is by preventing these molecules from being made. Let's talk a little bit about a thromboxane. I think I got a thromboxane here. I don't even know if I spelled that right. Thromboxane. These have roles in clot formation. They're named for their role in thrombosis. Potent vasoconstrictors, and they facilitate platelet aggregation. Let's see. What's that other one I got there? Leukotriene. This is a leukotriene. What do I say about leukotrienes? These are found in our blood cells, immunocompetent cells. So all of your like innate defenders, your neutrophils, your monocytes, your mast cells, the eosinophils, basophils. These guys are associated with the production of histamines and prostaglandins, which are mediators in inflammation, like we just talked about. So whenever these leukotrienes are overproduced, they may play a role in asthma or allergic reactions, and some of these compounds are actually the target for some of the asthma inhibiting medications. They inhibit the production or the action of the leukotrienes. All right, switching gears, let's talk about cholesterol. Cholesterol and some of the steroid hormones. So cholesterol is an important component of biological membranes. It's a precursor to some of the steroid hormones. Plants don't have a large amount of cholesterol. In fact, I don't think plants make cholesterol per se. They make phytosterol, which I have shown here. So let's talk about what I have here. So this is cholesterol. I can't spell. Sorry. So cholesterol has generated a lot of discussion. 13 people have gotten Nobel Prizes for this, for working on cholesterol. Cholesterol has a bad rap, and then it got a good rap, and then it's got a bad rap. Cholesterol is, more people talk about cholesterol probably than any other biomolecule. Okay. The brain tissue has about 10 to 15% of it, dry mass wise, as cholesterol. So cholesterol is important. It's everywhere. It's part of membrane flexibility. It's what lets membranes be flexible. Let me talk about that a little bit. So in animal cells, cholesterol provides the flexibility in our membranes that allows for the cellular movement that is actually in contrast to plant and bacterial cells, which don't have that need for movement. They have fixed structures. So cholesterol is made in many cells of the body. The liver makes the most. And from that, cholesterol can also go out to make some of the other hormones and some of the other fat-soluble vitamins. It's rarely found in prokaryotes, trace amounts in plants. Instead, plants synthesize this compound we call phytosterol. Cholesterol, very prevalent in the brain, synthesized by the liver. It's absorbed we can get it from our diet. We can also make it. And it has a role in membrane fluidity in addition to being precursors for many different compounds. So, like vitamin D. Okay. Let's talk about vitamin D real quick, since I've got that one kind of shown here. So vitamin D, the active form of vitamin D is known as, what is it? Cholecalciferol? Vitamin D3, essentially. Plays an important role in the intestinal absorption of calcium and phosphate. And thus, it plays a role in having some healthy bones. Technically, vitamin D isn't a vitamin. It's a compound made by the body. It acts more like a hormone, really. It can actually be made from cholesterol with ultraviolet light. So if we go out in the sun, we can make it with our skin. They started, I don't know if you knew that milk was fortified in vitamin D. This started because back in the early days of America, kids used to go and work in mines and stuff, and they weren't getting a lot of sun. And kids need more vitamin D than we do as adults because they're growing, right? They're building bones. And so because kids were part of the workforce or working in caves, mines, and that kind of stuff, the solution wasn't, hey, let's get the kids out of the workplace and get them in the sun where it's healthy. It's like, hey, well, let's just put vitamin D in their milk so that they can get it that way and keep going to work. It's funny. Let's see. Let's talk about. Actually, you can look up in the book about the vitamins. You can read about vitamin A. Read a little bit about vitamin D, vitamin E. I think vitamin E has a role for us as an antioxidant, and then what is it? Vitamin K. Vitamin K is used as a cofactor for enzymes, essentially. So, what about steroids? Let's talk a little about the steroids. Now, do I have any of those structures? I don't have any of those structures here. So while we're on the topic, let's just very briefly say that we have two different kinds of steroid hormones. We have mineral corticoids. These are steroid hormones that influence water and electrolyte balance. So like aldosterone is the primary mineral corticoid hormone. Though progesterone, which is another steroid hormone, does have similar functions. Aldosterone stimulates the kidneys to reabsorb sodium, to secrete potassium, and to reabsorb water. So this has the effect of increasing blood pressure, blood volume. Glucocorticoids are the other type of steroid hormone. These bind to glucocorticoid receptors that are found in our body, almost in every single cell. And these are used to basically like calm down an overactive immune system. So they reduce immune activity. And so there are various kinds. There are the progestogens, so also called the gestogens. These are steroid hormones that activate the progesterone receptor. And so the progestogens activate when they bind to the progesterone receptor. They have roles in pregnancy, have roles in the estrous cycle. Synthetic progestogens can actually go and, we know what some of these are for, used as birth controls, or they can be used, let's see, what are some of the other things that they're used for? Yeah, I don't know. I'm a guy. I don't have as much information as maybe you guys have. So there's the progestogens. There's the androgens. These are steroid hormones that bind the androgen receptors. These often stimulate development and the maintenance of male characteristics. Androgens are precursors of estrogens. We'll talk about estrogens in a second. Primary androgen is testosterone, but there are other variations of that. Estrogens, these are classes of molecules that have roles in the menstrual and estrous cycles. These are actually the most important of the female sex hormones. They activate the estrogen receptors, so estrogens activate estrogen receptors. These receptors in turn affect expression of genes, often as transcription factors. In the reproductive years, estradiol is the main estrogen, and then during pregnancy, the same thing, and then during menopause, it switches from estradiol. Estradiol is there are three main ones. I should have said that first. There's estrone, estradiol, and estriol. So estradiol is in the reproductive years. Then during pregnancy, estriol takes over. And then during menopause, estrone is the major estrogen. They all do slightly different roles. There are cannabinoids. These are groups of chemicals that bind to and have effects on the brain receptors called the cannabinoid receptors. And these actually can repress neurotransmitter release. These are compounds made by plants. Well, there are actually some made by our bodies called endocannabinoids. There are phytocannabinoids. Those are the ones made by plants, such as marijuana. And then there's synthetic ones as well. These endocannabinoids are also derived from the arachidonic acid. Cannabinoid receptors are actually really abundant. The largest number of what we call G proteins are cannabinoid receptors. All right. Let's see. I'm trying to think if there's any other of the lipids that I want to talk about before we move on to a new topic of transport. We don't need to talk about lipoxins. I mentioned heme, but we know what heme does. It's a primary component of hemoglobin. It's good for the transport of oxygen, CO2. Okay, that should cover our kind of examples. I mean, caffeine. That's a kind of a lipid. I mentioned we're not going to talk about all of them. We're not going to talk about waxes either. Okay, so let's switch gears. Let's talk about transport. So the cholesterol that's synthesized in the liver and the dietary cholesterol and lipids that we get from the large intestine, through absorption, need to be transported throughout the bloodstream to different tissues. And so there's a problem. They don't, hydrophobic and water don't really mix. And so the solution is that our bodies form these micelles. Remember micelles from when we were talking about soap? These micelles are packaged hydrophobic lipids inside a single layer of phospholipids with proteins on them called apoproteins that actually direct where they go. So these are like the addresses for excuse me, for these different. Well, we're gonna call these structures different things. We're gonna call them LDLs and HDLs and whatnot. So these lipoprotein complexes are actually combinations of apolipoproteins and lipids bound to them that solubilize fats and other nonpolar molecules, such as cholesterol, so that they can travel, to all the tissues that they need to go to. And these apolipoproteins provide the emulsification, so the mixing together so that they can dissolve in water, that they need so that they can actually, dissolve. They form these little tiny balls that are water soluble, even though the insides are full of non-soluble stuff. They carry all the fats, the cholesterol esters. So I should have mentioned cholesterol is actually transported as an ester for the most part. And then you can see here there are unesterified cholesterols, but those are incorporated into the membranes. And so we do see that. That's one of the main reasons that we want cholesterol is because of the membrane protein. But it gets transported as these ester molecules, these yellow ones here. Okay, and then fat-soluble vitamins as well get transported on the inside. So this is a list of some of the lipoprotein complexes. You can see that they all have the different names, kind of what they carry, what they transport. So the apoproteins are the distinctive features that are recognized by the cell receptors. So as these things are floating around, I actually have a different picture to kind of illustrate these differences. These are the apoproteins. You can see it here in blue. It's this big blue thing kind of wrapped around. These apoproteins are going to be recognized by the cells. The cells will then pull this whole globule inside it via something called receptor-mediated endocytosis. So yeah, we'll look at that right now. So this starts with, and of course we still need to talk about the different types of receptors. I think we'll talk about those right now. Let me go back here and actually give you the different types of receptors, because I should have done that. So we've got very low density. We've from biggest to smallest. We have the high density lipoproteins, the HDLs. I think I have something that is going to show you the composition of these in a minute, so don't worry. So they have high density lipoproteins. We have low density lipoproteins, LDLs. We have the intermediary ones, the IDLs. I don't know if you see any of those written here. Then we have very low density, VLDL, and then chylomicrons. Chylomicrons are the smallest one. Okay, so you can see here different apoproteins associated with the different complexes. Now, LDL is the main one, and we'll talk about its role a little later too, but this one transports cholesterol, or has the highest component of cholesterol. When an LDL is traveling around and a cell is ready to accept it, there will be an LDL receptor that will bind to the specific apoprotein. This causes something called endocytosis, which causes the cell to sort of engulf this little particle. Once it gets engulfed, it enters onto the inside of the cell, where it will be sort of digested. The receptors get taken off. You can see that the receptors, where are the receptors here? So the coating on this vesicle first gets taken off and recycled. The uncoated vesicle then gets sort of separated from the bilayer. The bilayer looks like it gets kind of pulled off, and they are able to collect all these receptors for, again, recycling. The receptors and that bilayer come back out on the cell. And then once this LDL is free, then something else, a lysosome, comes in, kind of breaks this apart and frees up all of the little cargo that was inside that LDL for use by the cell. Whether that's incorporation into new plasma membranes, or use of energy, or used for energy, that's what that does. Let's see. Let's talk a little bit about the different percentages of those HDLs and LDLs while we can. So HDL, known as the good cholesterol, is about 33% protein and about 30% cholesterol. LDL, or bad cholesterol, is only about 25% protein and about 50% cholesterol. So that's why, in terms of if cholesterol is a bad molecule, then it's the LDLs that we worry about because they have the most. There's the very low density. This carries triglycerides, mostly synthesized by the liver, and then chylomicrons. These carry dietary lipids. So these come from the absorbed food that we get, and so your intestines make these little chylomicrons for sending out everywhere. All right, so this is a chylomicron, by the way. Chylomicron with different apoproteins on it, so for different destinations. You can see all the little components on the inside. This is another image of a chylomicron, sort of showing it a little more realistically, where it's kind of been opened up for us, so we can see all the triglycerides inside the embedded apoproteins. All right. So chylomicrons and very low density lipoproteins deliver the triacylglycerides to the cells in the body. So both of these have high energy or energy rich triacylglycerides to deliver. So these are the best for getting energy to the cell. The triacylglycerides are actually stripped from the chylomicrons and the very low density lipoproteins through other proteins called lipoprotein lipase. So this goes and removes those fatty acids for use. And then the enzyme digests the fatty acids, the triacylglycerides, into fatty acids and glycerol. Sorry. There's like people doing yard work outside and it's making it hard for me to hear myself inside my head. Okay, so digests the triacylglycerides into these monoglycerides, and then those can diffuse into the cell much easier. So that's the chylomicrons. Sorry about the noise. Low density lipoproteins, these are the bad ones. So as the very low density lipoprotein particles get stripped of their triacylglycerides, they become more dense, okay? They become more dense. So this is from the intestines, right? From the liver to the cells. Lipoprotein lipase, we just talked about that, triacylglycerides to the cell. Now, as they lose those, they become more dense, and so they go back to the liver. They go back to the liver where they get repackaged and remodeled as LDL. So then they come back out as LDL. Now, LDL delivers cholesterol to the cells where it can be used. So cholesterol, not triacylglycerides. Cholesterol. So cholesterol to the cell where it can be used as membranes for the synthesis of hormones, whatever. Cells take up the cholesterol by the receptor-mediated endocytosis. We looked at that. The LDL then binds to the specific receptor, gets internalized, right? Receptors recycled, all of that good stuff. Okay. HDL. Let's talk about HDL. HDL is cholesterol scavenger. So the HDL precursors, they actually get synthesized and secreted by the liver and the small intestine. So they're not showing the small intestine ones here, but HDL. So in circulation, they're running around gathering cholesterol to form mature HDL, which then returns the cholesterol to the liver via various pathways known as essentially reverse cholesterol transport. So the job of HDL is to run around collecting extra cholesterol. If you've got it, it's like a trash man, brings it back to the liver. So it can be repackaged as LDL to go back to the cells. It's like cleaning up the streets, right? Cleaning up the trash. Now the liver, in addition to also packaging it up as LDL, also excretes it into the small intestine as bile salts, converts it to bile salts, which help emulsify fats from your diet. So there's a reason this gets stored in the gallbladder, the bile salts. Bile salts also come from cholesterol. So that's the HDL pathway. Now, here's another kind of picture of that. So here's cells getting cholesterol. Here's fat cells, right? If we're going to store fat, can store it like that. That's what fat cells look like. All right. So I was going to say something. Oh, I'm trying to set up a big picture here of why this is unhealthy and what the problem is. Okay, so we know what everybody's job is. Chylomicrons bring dietary lipids for energy or for for storage or for energy. Very low density lipoproteins too transfer these things. They get repackaged as LDL to take cholesterol to the cells. If there is any excess cholesterol, we're going to talk about what happens. We haven't talked about that yet, but we know that LDL, once it does the job, gets takes cells to the takes lipids to or cholesterol to the cells, then it can return to the liver for getting for more packaging, and then HDL runs around scooping up extra cholesterol. Okay, so what happens if it builds up in the blood? Sorry about that. My graphic got cut off. Okay, so if we have lots of LDL in the blood, what actually happens is it stops the production of LDL receptors in the cells. The cells go, hey, stop. We don't want any more cholesterol. There's way too much cholesterol. So now that the fact there's so much cholesterol in the blood, I'm not even going to make LDL receptors anymore. And so all the little receptors that were out here recognizing the LDL, they're gone. So now this stuff starts to just build up out in the blood. Okay. LDL is bad because it has lots of cholesterol in it. HDL is good. HDL runs around scooping up LDL, kind of, helping. Sorry, I lost track of what I was going to tell you guys. Okay, so the liver is also important for modulating levels of lipids. Okay, it uses special LDL receptors on its surface. That was back here to get LDL back in. So the liver LDL receptors actually bind to the LDLs that were not taken up by other cells. So if you couldn't find home in a cell, you're floating around looking for a home, the liver takes you, okay? Pulls you back in. And so again, high levels of LDL are a signal to the liver to actually stop making very low density lipoproteins, to stop, doing this, because there's obviously no need for all of this for these fats. So then that leads us to this picture. So the liver is already going to have to stop, because this is building up. So the liver is already going to start regulating how much VLDL comes out. But also the cells themselves stop trying to accept the LDL. So now we've got lots of LDL. Okay. Damaged LDL or extra LDL might get taken up by the immune cells called macrophages if they don't just deposit all their extra cholesterol all over the insides of your arteries as plaques. So that's one thing that can happen. The cholesterol just gets deposited. The other thing that happens is that these immune cells get jammed up with these macrophages that are taking in damaged LDLs. And you might wonder why damaged LDLs happen. Well, this happens from reactive oxygen species, radicals. So antioxidants is a thing that your body tries to fight, but oxidation happens. And so antioxidants or oxidation of LDL leads to damaged LDL, and damaged LDL gets taken up by macrophages. These immune cells get jammed up with cholesterol, they die, and then they just deposit on the walls of the arteries. And then this is what leads to atherosclerosis, having the buildup of all of this plaque essentially on the inside. So if HDL is high, it can actually scavenge the cholesterol from the overloaded macrophages before they die. So having high levels of HDL, low levels of LDL tend to be in line with what you want to do to kind of stay healthy. And again, they often, what are ways of kind of reducing the amount of cholesterol? Well, just ingesting less of it is going to have a role because you make it yourself. But if you ingest less of it, then you shouldn't have as much of an issue. Okay. Let's talk now about lipid bilayers. Excuse me. So, every cell has a cell membrane, plasma membrane, composed of a lipid bilayer, phospholipid bilayer. Eukaryotic cells also have membrane-enclosed organelles. Inside their cells, they also have other little organelles that also have these lipid bilayers. Polar head groups in contact with the aqueous environment, non-polar tails buried within the bilayer. And so the major driving force for the formation of these bilayers, of course, is hydrophobic interactions. So outside of the cell, inside of cell, and then impermeable, well, not impermeable, semi-permeable, because we're going to talk about how we actually permeate this. So hydrophobic molecules all sort of kind of live in the middle, and the hydrophilic molecules get attracted to the ionic environment created by these phospholipid polar head groups. But molecules can't just pass through the bilayer. They get either associated to one side or the other, but because of their charge differences, they can't move through. So this is going to lead to the different ways that we transport molecules, the different ways the body has figured out how to transport across these membranes. But we'll talk about that in a little bit. The types of molecules that actually can get through are going to be neutral ones. So water, carbon dioxide, carbon monoxide, oxygen. Anything that's polar is not going to be able to get through this membrane. So we want this. We want an orderly movement of compounds in a cell. It's crucial for the cell to be able to get food for energy, to export materials, especially wastes, to maintain osmotic balance, which is having equal osmotic pressure. We'll talk a little bit about that towards the end. Creating secondary gradients or creating gradients, either pH gradients or concentration gradients for energy production or secondary transport. We're talking about the things that are benefits of being able to maintain what crosses across the membrane. Provide electromotive force for nerve signaling in terms of our potassium and sodium pumps. Store energy in electrochemical gradients for ATP production. I mentioned that. pH gradient. Even photosynthesis has this kind of stuff. Is related to the fact that we have charges that separate across these membranes, and that we can selectively move certain molecules across and not others. All right, so we're going to have to talk about the types of lipid composition. So that's what I wanted to show you here. So we actually have an unequal distribution of phospholipids within the lipid bilayer. Now, this is mostly fixed. You can see on the inner layer or the inner leaf versus the outer layer, we have a lot more of the phosphatidylethanolamine. On the inside, the phosphatidylserine, phosphatidylиноцетол. On the other side, on the outside, we have the phosphatidylcholine, the sphingomyelin. So this is what we talk about when we say the unequal distribution of the phospholipids. Now, phospholipids cannot easily cross from one side of the membrane to the other. We call that flip-flop. And I'm actually going to show you some of the enzymes involved in getting that job done. Lateral diffusion, which is moving side to side or moving around within the membrane, is possible. And I am going to show you some of that. And we did talk about how when some of these flip, like phosphatidylserine, when this thing flips to be more present on the outside, that actually indicates the cell death. Okay. So I want to talk about this diagram that I've got here, and I don't know why I've got it here. Oh, okay. The distribution of the lipids, percent membrane lipids. So I was talking also about the organelles on the inside of the cells. Sorry. The organelles also contain lipids, and they also have phospholipid bilayers. So this is some of those compositions as well. So this is just showing percentages, not which side of the membrane they're on, but again, just so you can see that the compositions of some of these are quite different. Lysosomes, for example, contain a lot more sphingolipids. Plasma membrane lipids have a lot more cholesterol in them, and some other differences. The nucleus actually contains no cardiolipin, whatever that is. We didn't talk about the lipins. Okay. Um, um, it is interesting, though, that the cardiolipin is almost only found in the mitochondrial membranes. You see how much it is there. Um, Anyways, okay. So just to kind of show you, we did talk a lot about these different phospholipids, and just to show you that they do have some role in binding certain proteins to the membrane. We haven't looked at that yet, so just keep that in mind. That some of the differences in, like, let's say the amino alcohols are going to be their charge, and that charge is going to help attract or repel various other molecules and kind of recruit them to either bind there or not bind there. Okay, let's talk about lateral diffusion. Lateral diffusion's coming. This is my slide on, what was I going to talk about here with this? I guess I'm just showing the different types of protein associations with the membrane. So you can see here in the membrane we have the inside and the outside, different types of proteins. So I've got integral proteins here. These are those that span the entire membrane, the entire bilayer. So they're visible on both sides. We've got peripheral ones only interact with one side or the other. Associated proteins are proteins that actually don't attach to the bilayer at all. They attach to proteins that are attached to the bilayer. So either those peripheral or integral proteins. Anchored proteins, these are those that can be sort of part of their, remember we said that some of the fatty acids can serve as anchors. These fatty acids can very easily go and embed into these lipid bilayers. But I did want to comment a little bit more about the molecular organization of this lipid bilayer. We'll talk about, let's see, anchored. I mentioned anchored peripheral, mentioned I didn't mention these transmembrane domains. Let's talk about these. So down here, I've got transmembrane domains. So these are the things that hold these integral proteins into the membranes, these lipid bilayers. So many of the integral proteins are anchored in place using single alpha helices. So this is an alpha helix right here. About 25 to 20 amino acids pass through this membrane, and they're mostly nonpolar and hydrophobic amino acids. So that's how this protein sort of just gets into this lipid bilayer. Other integral proteins actually pass the membrane many times, like up to 17 times, with what they call alpha helical bundles. Others form beta barrels. And so I think this is a beta barrel. This might be an alpha helix bundle. But again, these are the kind of domains that we see in proteins that are integral. These are the transmembrane domains, TMDs. This is what I wanted to talk about in terms of lateral diffusion. So we think of the plasma membrane as being something called a fluid mosaic. In fact, if you Google fluid mosaic or YouTube that, you should find a nice video that kind of shows the little, if we go back to this, shows all of these proteins sort of wiggling and moving around. Every one of these phospholipids too can shift and move around, and we know that because if you were to take a top-down view at part of the plasma membrane and then with light actually stain some of those lipids and then study that for a while, you would see that those color changed ones diffuse away, they move, meaning that at first they're all right next to each other in the bilayer, and then they start to wiggle away from each other. You see they're spreading out, and this happens. They're free to move laterally. So any membrane protein, integral protein, cholesterol molecule, sphingosine molecule, any one of them can move side to side, forward, backwards, left and right, around and around. They just can't flip-flop to the other membrane. Well, they can, but they require special enzymes like flipases, flopases, and scramblases. So a flipase will get you from one side to the other. A flopase looks like it, what's the difference? Just takes you from the inside to the outside? Or, yeah, I'm not really sure. I don't really know about the flipases and the flopase method of action, but I know that there are a couple. So flipases move membranes, phospholipids from the outer leaf to the inner. So there we go, outer to inner for flipases. Flopases move inner to outer, and scramblases will move either direction. So they can move outside to in, inside to out. Okay, so I lied. I did know. Cholesterol. Let's talk about cholesterol. So cholesterol actually has an effect on fluidity. I may have mentioned that a little earlier. So cholesterol actually increases the fluidity of the phospholipid membrane by disrupting the packing of the alkyl chains. So if you allow the alkyl chains to pack together really well, we know that that actually raises the melting point and it actually makes it more rigid. So by inserting cholesterol molecules and breaking up that packing, we get a few more, we get more fluid character, we get some more wiggle room. And so what cholesterol does is it doesn't change the melting temperature or the midpoint of that transition from solid to liquid, but it widens the range. And so it actually can provide some more stability. If you remove cholesterol, you can add more flexibility if you add cholesterol. So it's kind of like a buffer. Okay. So we're going to talk about different types of transport. Now, there's diffusion. And this is for CO2, carbon monoxide, oxygen, water, not really water so much, but most non-charged, neutral, non-polar substances will diffuse pretty easily. Diffusion happens right across the membrane. Then we have facilitated. Diffusion. I don't know if I spelled that right. 2 S's? No S. 1 S? 1 S. Facilitated diffusion. This involves having a protein. Now, a transporter protein can be two types. There can be an ion channel, and this speed is roughly the rate of diffusion. So if there's a high concentration gradient on the outside, then it's going to go fast. If it's a slow concentration gradient or not a big difference, then it might go slow. But this is a fast type of transport that requires no energy. There's a slower one, a slower facilitated diffusion, and that involves a transporter protein. And the transporter protein may have to bind a molecule first, and then conformation change happens, and then it'll spit it out. These are much slower. Both of these, though, require no energy. Okay? So sometimes binding of the proper molecule causes a conformational change that results in like a flipping, and the open side closes, the closed side opens, and the molecule moves. Okay? Then there's active transport. Now, all of the methods that we've looked at just now require no energy, but with active transport, energy is required. So here's an example of that. So let's take, for the example, the hydrogen. I'm sorry, I always say hydrogen. This is the sodium-potassium pump. So this thing effectively moves sodium outside the cell and takes potassium inside the cell, both against their concentration gradient. And the way that this one works is we've got this protein, so it binds some sodium, and then ATP is hydrolyzed so that this could be phosphorylated. So now this molecule gets phosphorylated, this protein, changes its shape. So now it changes shape, and the sodiums from the inside got pumped out. But now it's open facing outward. So some potassiums go in. And then something comes in here and removes the phosphate group, right? Cutting off the phosphate group is going to cause the shape to flip back. And now the potassiums on the outside go in. And then this cycles. But adding that phosphate group requires the ATP. And then there's one other. So if it requires ATP, then it's called active transport. And if it requires ATP if it doesn't require ATP, but it requires some kind of, what's the word, concentration gradient or some other kind of gradient, in this case, like a hydrogen gradient, then it's called secondary active transport. And so what happens here is that there's some kind of energy or some kind of, something that's happening over here that's causing hydrogens to be pumped across the membrane. So high concentration of hydrogen ion in one part of the membrane, low concentration in the other. And so now hydrogen wants to just, passively go along its concentration gradient. But the transport of hydrogen back into the cell actually facilitates lactose being brought into the cell. Like lactose can't get in without the hydrogen ions. So in this case, we're using this proton gradient to drive the transport of a different molecule. That's called secondary active transport. This is important too. Both of these, not just the sodium potassium, but also anything with transport across the cells for maintaining proper electrolyte balance. Water has a tendency of wanting to dilute compartments, dilute systems. And so if there are too many ions or too much concentration of something on one side of the cell, water's going to want to try to go there. And this can have some serious physiological effects. We have, let's say, red blood cells. Our red blood cells are these little like donut-shaped kind of concave cells. If the solution inside is much more concentrated than the outside solution that the cell is in, well, then water is going to start to flow inside the cell, leading to a swollen cell that may burst. And we call this a hypertonic solution. Or we would say that the solution is hypertonic to the cell, meaning hypotonic to the cell. The solution itself has a way less concentration, hypo, than the inside of the cell. We would say that the cell is hypertonic to the solution. Vice versa, this could have a much lower concentration than the outside environment, in which case the solution is hypertonic to the cell, and this is going to cause the water inside the cell to try to escape and to dilute the outside environment, leading to the blood cells shriveling up or crenating. And these, of course, also break. So red blood cells is a nice example, but this is true of any cell. So electrolyte balance is important. Water follows the ions, and this is how we get water to move around in the body. Control the ions, control the flow of water. Okay, I think that's it for the information on lipids. I can't think of anything else at the moment that I wanted to talk about. There's obviously a lot more information about differences in active transport and the different types of facilitated transport, but that's not the goal of this class. The goal here is to expose you guys to the fundamentals, not to go over all the differences. Blood types. We didn't talk about blood types much when we were talking about the different anchored membrane proteins. Just membrane proteins in general. They can be, remember we talked about they can be integral and span the entire membrane. They can be, and of course these integral ones are often used as docking sites for attachments to extracellular matrix or for other cells. I mean, for holding cells together. Receptors in the signaling systems or just transport of molecules. There's peripheral membrane proteins. These are the ones that are just kind of stuck onto one side or the other. These guys have roles in, what do they have roles in? I can't think of a single role. We'll talk about some later, probably when we get to one of the later chapters. Interactions with proteins or molecules. I mean, ribonuclease I know is one, but right now I couldn't tell you what ribonuclease does. And then there was the anchored proteins, and the anchored proteins are just sort of embedded in the lipid bilayer by like a fatty acid. These are like our blood types. Our blood types have different embedded proteins on the outsides of the. So we have glycoproteins embedded on the outside of our blood cells, depending on which type of glycoprotein determines what kind of blood type we have. Okay. Anyways, the book has some sections on blood type you can read if you're interested in that. And then again on the vitamins as well. But other than that, guys, this was a long one, but hopefully it was something that you could follow. Okay, then take care, and I will see you guys next time.